Hello students, I, Mr. Vishal Shah, Assistant Professor, Krishna Institute of Pharmacy, Karad, once again welcome you all to this yet another session of chapter Fats and Oils. Today we are going to discuss about the remaining part of the compound lipids. In previous videos we have discussed about the classification of compound lipids. In that we have started with the phospholipids. We have covered a few types of the phospholipids like phosphatidylcholine, serines. Today we are going to start with the remaining part of the compound lipids. So let's start with our today's session. Before starting with our today's session, we will see what are the learning outcomes of this session. After this session, students will be able to explain classification of compound lipids, uses of the different compound lipids and different types of compound lipids that are present in our body. So our today's first compound lipid that we are going to study is phosphatidic acids. These phosphatidic acids are the forms of phosphatidates, which is a part of phospholipid. They are the major constituent of the cell membrane, and phosphatidic acids are the simplest diacyl glycerophospholipids. The structure of this particular phosphatidic acids like this. Also phosphatidic acid consisting of a glycerol. Here if you look at in this structure, this is the part of the glycerol. The saturated free fatty acid that is bonded to the first carbon atom, then unsaturated free fatty acid that is bonded to the second carbon atom, and phosphate group that is bonded, or phosphoric acid you can say, that is bonded to the third carbon atom. So this is the structure of phosphatidic acids. Here you can see there is no presence of any head alcohol group is present. That's why also they are having a negative charge, that is they are present in the form of the cations. So this is about, anions. So this is about the phosphatidic acids. Then second type that we are going to discuss today is phosphoinositates. Phosphoinositates are found in phospholipids of brain tissue. So whatever brain tissues are present, the major component of these brain tissues is phosphoinositates. Now, one of the major difference between phosphoinositates and the whatever the compound lipids that we have seen, the phospholipids, they are having the, or glycerophosphatides, they are having major difference in presence of the glycerol that is present as an alcohol. As in the classification of phospholipids, we have seen that phospholipids are classified into glycerophosphatidates, then phosphoinositates and phosphosphingosides. So this is the second class of the phospholipids, that is the phosphoinositates. Now the main diet source of this phosphoinositates is soybean. It plays an important role in transport process in all the cells. These are the phospholipids where cyclic hexahydroxy alcohol, which is known as inositol, replaces the base. The inositol is present as a stereoisomer, which is known as the myoinositol, and on hydrolysis they yield 1 mole of glycerol, 2 moles of fatty acids, 1 mole of inositol, and 1, 2 or 3 moles of phosphoric acid. Means in case of phosphoinositates, head alcohol group that is present, it is replaced by the inositol. So this inositol is nothing but the hexahydric alcohol. The structure of commonly present phosphoinositates are as shown over here. You can see here there is a presence of, here again, the glycerol, where first carbon atom is connected with the free fatty acid, Here also there is a presence of free fatty acid, then phosphoric acid group, and to this phosphoric acid group there is a presence of hexahydric alcohol. Hexahydroxy alcohol is present. Also, whatever these alcoholic functional groups are there, they may be converted into the again phosphoric acid derivatives. So depending upon these, phosphoinosilicates can be of different types like mono, di or triphosphoinosilicates. So this is about the second class of the phospholipids, that is the phosphoinosilicates. One of the common example of this phosphoinosilicates is lipositol. Lipositol is the example of the commonly found phosphoinosilicates in the humans. Then after phosphoinosilicates, next class of phospholipids is phosphosphingosides. These phosphosphingosides are also known as sphingomyelins. These are the commonly found in nerve tissues, especially in the myelin sheath of nerve fibers, and that's why the name is also given as sphingomyelins, and apparently lack in plants and in microorganisms. So main difference between the earlier class, here you can see in this structure, instead of the glycerol there is a presence of the sphingosine. So here glycerin is replaced by the sphingosine, which is a alcoholic functional group that is present in all the phosphosphingosides. Here you can see the structure of sphingosine, where there is a presence of this particular long chain acidic functional group. Here you can see the primary alcoholic group is present. Now when we carry out the hydrolysis of these sphingomyelins, it will be converted into choline, then phosphoric acid molecule and the sphingosine molecule. Now this sphingosine molecule is consisting of this long chain fatty acids, which is comprising of the structure of sphingosine. Further, in a syndrome called as Niemann-Pick disease, the sphingomyelins are stored in the brain in large quantities. Means if in the brain these sphingomyelins are stored in large quantities, that leads into the syndrome which is known as the Niemann-Pick disease. And in this disease, concentration of these sphingomyelins is more in the brain. Now these differ from other phospholipids in their lack of glycerol. As I said, in the structure of these sphingomyelins, glycerol is replaced by the sphingosine, and the presence of another nitrogenous base, that is the sphingosine, or a closely related dihydrosphingosine besides choline place of the glycerol. Choline is present, and in addition to that choline there is a presence of sphingosine or the dihydrosphingosine. Now sphingomyelins are electrically charged molecules and they contain the phosphocholine. On hydrolysis, the phosphosphingosides yield equimolar amount of fatty acids, then phosphoric acid, choline, either sphingosine or the dihydrosphingosine, but no glycerol, because in the structure of sphingomyelins there is absence of glycerol is there. So due to after hydrolysis, glycerol is not obtained. Thus in these compounds the atomic ratio of nitrogen to phosphorus is 2, in contrast to phosphoglycerides, where this ratio in case of phosphoglycerides is equal to 1, or you can say unity. Phosphoinositides, however, do not contain a nitrogenous base. After this, next type of compound lipid is glycolipid. Now we know that after phospholipids The second class and one of the major important class is glycolipids. Glycolipids are nothing but the lipids in which sugar is present as a non-lipid portion. That's why the name is given as glycolipids. Further, these glycolipids are also classified into two classes. One is the cerebrosides, which is also known as the glycospingosides, and the second class of glycolipids is gangliosides. Now, one by one, we will discuss these glycolipids. One is the cerebrosides, which is also known as the glycospingosides, and another one is the gangliosides. First, we will discuss cerebrosides. Now, the cerebrosides are important constituent of the brain, and approximately 8% of the solid matter that is present in the brain is made up of these cerebrosides. These may also occur in tissues other than brain, since the head group characteristically consists of one or more sugar units. Cerebrosides are also known as glycospingosides. Now, this is the common structure of the cerebroside, where you can see that there is a presence of galactose as a sugar moiety that is present, along with a sphingosine molecule, and which is also consisting of the fatty acid component that is present in the structure of these cerebrosides. Like phospholipids, glycolipids are also composed of hydrophobic region containing two long hydrocarbon chains and the polar region which now contains one or more sugar residues. As you can see here, there is no any phosphate group is present because they are not phospholipids, they are as glycolipids. The hydrophilic portion is made up of the sugars. Both phospholipids and glycolipids form self-sealing lipid bilayers that are the basis of the all cellular membranes. These are present in much higher concentration in medullated than in non-medullated nerve fibers. There is evidence that they are also occur in some plant organs. The structure of cerebrosides is similar to that of phosphospingosides. They contain high molecular weight fatty acid, sphingosine, and either galactose or glucose instead of choline, but no phosphoric acid. They have no electric charge since their polar head groups are neutral. In general properties, they resemble the sphingomyelins. So this is about the cerebrosides, which are also known as the glycospingosides. So here we have completed with the cerebrosides. Now in next video we will discuss about the gangliosides. So up to this point we have covered with the phospholipids and we have started with the glycolipids. We have seen one class of the glycolipids, that is the cerebrosides. Now you have to go to this particular website and use this particular code to give the answer of this question. Phosphosphingosides are also known as dash 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 dash. Thank you. If you like the content of this particular video or this channel, you can subscribe to my YouTube channel. You can share with your friends, and if you have any queries related to the pharmaceutical organic chemistry, you can contact me at this particular email ID. Thank you very much.